Morning, Paul. This is Beth. I was attempting to type up this popsicle stick experiment, and I realized that it would be much easier to just show you. So I have some makeshift popsicle sticks here. I'm going to use some straws instead of popsicle sticks, and just kind of talk you through this exercise 6 in the lab manual and what we want the students to be doing. So I have blue and orange popsicle sticks here. Instead of blue and pink, but we're starting out with just one of each of the popsicle sticks. So we have one long blue one long orange one short orange one short blue one. Let's make sure I can get this all in the camera. Sorry, technologically impaired this morning. So move this a little closer. Okay. Oops, I just put that on top of my lab manual. I need that. Okay, there's the popsicle sticks. So, at the beginning of the activity, they need 2 of each, but they just start out at the very first with 1 of each, representing a single chromosome. So just 1 copy of the genetic material. These 2 long ones are meant to be a homologous pair, so they're 2 copies of the same chromosome, 1 from your mom, 1 from your dad. And same thing with these short ones, they're meant to be homologous. So we have 2 homologous pairs for a total of 4 chromosomes here. And that's their first question: how many chromosomes does your cell have? So they should identify it as 4. Then their next instruction is to place duplicate sticks side by side on top of the originals to represent replication. So now they're going from being single chromosomes to duplicated chromosomes. So since I only have straws, then I'm just going to set these other halves next to it. But you can envision these would now look like duplicated chromosomes or 2 popsicle sticks. So here I would always ask students again, how many chromosomes do you have? It's not a question in the lab manual, but here is where a lot of students will say 8 now instead of 4. So that's a good place to point out a misconception. So then, now place homologous chromosomes next to each other to show the synapsis that occurs during meiosis. So unfortunately I just kind of already did that. The homologous chromosomes are the ones that are both long and both short. So they move in the cell so that they're next to each other during prophase 1. So then it says to place them next to each other. This represents metaphase 1. So we would envision that they're all lined up along the middle of the equatorial plane. Draw a sketch of your chromosomes in the following arrangement. So here we would just be looking for them, drawing the homologous pairs next to each other. Now separate the homologs from one another. This is anaphase 1. And assemble the sticks as they would appear in telophase 1. So this is really the part that students mess up on most often. Because in anaphase 1, the homologous pairs separate from one another so that one duplicated chromosome moves towards one end of the cell, and the other duplicated chromosome in that pair moves towards the other end. And same thing with the short ones. So we would end up having 2 separate nuclei that each only now have 1 long duplicated chromosome and 1 short duplicated chromosome. Same thing here. So if we imagine that there was a divider here and these were 2 separate nuclei, then those would be the 2 nuclei that result after telophase 1. So if you're following along on page 128 also, it shows all the phases and where the chromosomes should be located and how they move apart during anaphase. That's kind of the tricky part. So now they're supposed to describe the chromosomes in each daughter cell, so they should describe 1 of each chromosome. So now they're called haploid cells because there's only 1 copy of each chromosome. And it asks, do the nuclei in telophase 1 still have homologous chromosomes? No, now they're in 2 different nuclei. So if you have done the simulation correctly, your cells still have replicated chromosomes, i.e. there are still 2 sticks of each size and color that must be separated from one another. So now we have to go through all those phases of meiosis again. Prophase looks a lot like telophase 2, so nothing much changes. Metaphase, they line up in the middle, so we could sort of imagine already that this is the middle that they're lined up on. And then in anaphase 2, now it's finally the duplicated chromosomes that get pulled apart. So each arm of this duplicated chromosome we call a sister chromatid. So now these sister chromatids will get pulled apart to opposite ends of the cell, so that finally our 4 daughter cells each have one long chromosome and one short chromosome, and they're single copies instead of duplicated copies. So that would represent 4 different daughter cells, each having one long one and one short one. And another thing that I like to point out to the students is that the segregation could have occurred in a different way, so that one of the daughter cells actually had a different color arrangement, so we could have resulted with this as well. It's just kind of luck of the draw how it works out. So that's pretty much the popsicle stick simulation in a nutshell. Again, I think the biggest thing there is during anaphase 1, making sure that the homologous chromosomes separate from one another, thus reducing the number of chromosomes. So hopefully now I can figure out how to get this thing online so that you can watch it, and hope you're having a super Monday. Talk to you later.